Alright, we have a really special lab here for Station 2 of the plant pathology lecture for TapRoots. Essentially, students will be discovering nematodes as a sign of plant diseases. So one of the signs that they'll be kind of investigating are root knot nematodes, which cause this root ball to form here. And follow the pointer here, but essentially, kind of zoom in, we have the roots. It's bad quality, but you're looking for cankers. Those are the infection sites of the nematodes. And again, you'll see kind of the nodulation here, which is a result of the infection site. Where the root thickens is where the nematode has infected the root. As it narrows, that's the uninfected root. Now they're going to be able to contrast this with a couple of petri dishes with pre-labeled roots. Here, pardon me for a second, but this is a tomato root that has not been infected. There we go, this can see it better here. And you can kind of see it looks healthy, very thin, very consistent thickness. But if we go to a tomato root that has been infected, the contrast is apparent. Again, the cankers are forming there as a result of the nematodes infecting the site. And then the next thing I kind of want to focus on is the nematodes themselves. We're going to have two different types of nematodes in culture. Here's a box full of goodies. Now it's really important that we respect the supplies here, because again, this is lent out from the nematology lab from USDA. Much appreciated from Carol and Susan over there, as well as Shannon. But this you can essentially just put right under the dissecting scope. And this is actually, they refer to as a beer nematode because they like yeast. It's all pre-labeled, but you're going to be able to see when they look into this, the dissecting scope, they're going to see the nematodes swarming around very vigorously. The ones that are still are actually the dead adults, and ask them essentially look at the size differences and so forth. Now we have another slide of free-living nematodes. Now this was just set up today, so later on in the week they're going to see eggs as well in the solution. So this is a couple of visual aids to help out with this lab. And if you so choose, there are depression slides in here. They're not required for the lab, but if you wanted to extract some of the beer nematodes, as we can simplify it there, you can pull it from this vial here with these extraction devices. But that's up to you. Okay, there's that. And for your own benefit, we can pass these around. Here are some worksheets that accompany the lab, and some other visual aids as well. Dissecting scope is very expensive. Again, it was lent out by the nematology lab at USDA, so please treat that with respect. Please, please, please. And yeah. Oh, another cool thing. So the root knot here, nematodes, they have eggs on the roots here on this root mass. So if you add just a little bit of water and you let it sit in the petri dish, you're going to see them hatch, most likely. If you have time, that might be a kind of a cool thing to do. So good luck, and hopefully you enjoy it.